Rolling this way.
Yeah, so much for our genome, exactly. So I should just say, I was asked to do this, and I said to George, I'm not too sure I like this whole thing, and I'm not sure I'm smart enough to, so I said, could I ask for help? So Carl works at the New York Times and works at home and works at every magazine in the world seems to have his work. So I asked if we're going to do this as team, tag team, I just thought I'd just get Carl as my partner. So this is Carl Zimmer. You read the New York Times. He's in it all the time. He has a website called The Loom, which is a wonderful one. He's fascinated by parasites. Anything else?
What more do you need?
Yeah. Okay.
This is Robert Krulwich of NPR, and if you haven't heard his show Radio Lab, you really should go and listen to it now. It's an excellent show.
All right, now, so the little chair here, this is just deeply odd, because we're going to put the king in the little chair, but that's because we just got the big chairs. So, Jim, could you just take the little chair? And this will be one of those rare experiences where the reporters can be taller than the Nobel guy. So, 1953, you created the Human Genome Project that has led to this and that and the other, but for
53, double helix.
Double helix. Yeah, so that would be 86 was the genome?
We talked about it. It sort of started in 88, I think.
88. So far I'm wrong with every presumption. What about the decision you made very early on to be personally sequenced? You're one of the very, very first. Why did you do that? Other than to be fabulous, I suppose.
I don't remember.
Thank you. You may sit down.
When Jonathan Rothbard asked me to do it, I thought, we had to do someone. And so at this stage of my life, I tend to say yes.
Were you worried about anything?
No, I said I didn't want to know the apolipoprotein E, whether I had the 2, 3, or 4 variants.
For Alzheimer's?
Yeah, because one of my grandmothers had it, so and she got it in her 90s, so or probably had it before.
So did it come with a little black sort of? Did it it's like you can't look under this line kind of thing?
No, we supposedly took it out, and I don't want to go into it because it's more complicated.
Okay.
Of whether you can, from haplotypes, determine things, even though you take
But did it
out.
bother you that somebody in the world might know something about you that you don't know? Did that?
No.
Okay.
No, it's partly my age. If I were 20, I would probably,
Want to know?
Well, yes.
Why? What's the difference?
Well, because you can just worry. And at 80, I'm not worrying about, whether I'm prone to cancer or something like that, which, I could have worried about when I was 20. And, I had a skin cancer at a very early age. So, and then when I went to down in Houston and Baylor, they told me, I had mistakes in a couple DNA repair genes. And the actual advice, they were wrong in 2 of the cases. So the advice was, luckily, because one of them was, I was supposedly homozygous, and then, so I didn't think about it, because, I sort of should have cancer. And
you sort of should have cancer because your dad died of lung cancer.
No. This was because the chief reason you get cancer is because you can't repair your DNA, and so BRCA1 is terrible, or BRCA2 because they're DNA repair genes, and Bloom syndrome. There are lots of.
When you looked at what
But anyway, someone at Edinburgh told me that I was one base pair away from the one they knew was bad. So well, it was, but I didn't think about it, and because I have other things to think about. I think that's probably the chief reason. Other more immediate worries.
Were you surprised by anything you read? Like, there it was. There you were.
I've got two things which are useful. One is, I'm partially lactose intolerant, and I guessed that with time. But but Baylor didn't consider it important, so they didn't tell me. And then Craig Venter's lab compared his sequence and mine with regard to our cytochromes, whether the enzymes which metabolize drugs. And then I learned that I was a very poor metabolizer of beta blockers. And I would have been a very poor metabolizer of antipsychotics. And
so this is like, did he do like some kind of celebratory dance around you called My Beta Blockers Are Better Than Beta?
No, but they'd given me and put me to sleep. And now, so I realized instead of taking one a day, I should take one a week. And, I bought a machine to measure my blood pressure and saw how long,
Oh, so you used some of this information?
So, no, it's very useful. So, to the extent that my blood pressure is controlled, really was helped by it.
What about
I was helped by Craig Venter because he was interested himself, and he's perfectly normal in it. Whereas, some people are very fast metabolizers, some are slow. It explained a long mystery. I have a son, and don't write about him. And I really say that because it's not.
There are a lot of people here, by the way.
I know, but it's not kind. He doesn't like to read himself, being schizophrenic. And once he was given an antipsychotic because he, hit a psychiatrist, which are generally a justified act. And crazy. And so he went into neuroleptic malignant syndrome, of high fever, almost died. And that was because he got one of my two bad metabolizing. And I just thought, they didn't like him and just gave him a lot because they, really were afraid he was badly psychotic, whereas the doctor was psychotic, actually.
But wait a second. What about the question?
So now psychiatrists do worry about whether you're a good or bad metabolizer of,
they get decked by the patient.
Well, your patient can die. So it's and so I'm, I think there could be a company, founded or eventually be bought by one of the big guys, just to do the cytochromes. And so your doctor knows what things you will metabolize and don't when you go to a cardiologist or any other. It's important.
Well, let me ask you two burdens that occur to me. As a parent, now knowing that you are the author of a defect in a kid and it's assigned to you, does that complicate the idea of fatherhood for you?
No, not at all. Why should it?
Well,
that's an absurd question. Why should it complicate?
It's my specialty.
Yeah, I know, but it is really, it's just genetics.
So that's just the way it goes. All right.
Yeah, that's all.
What about this sentence? We will be able, within 10 years, to diagnose the problem of schizophrenia by looking at a patient's DNA. You said that. Do you still believe it?
No. I don't know what year I said that, but now it seems likely that every family will have their own specific defect. There's going to be thousands of different ways you can get schizophrenia. There are a few hot spots where there might be, if it's on 15, 16, a couple of percent you may be able to. Otherwise, you just mess up the way synapses function. And you can do this in a very large number of ways. We don't understand why so many proteins are involved in synaptic function. There are about 1,000 in the postsynaptic density alone. So there are many different ways in which you can go wrong. And
so then what will being able to see these genomes, how will they be able to help?
Some way you will hope that they'll put together to some pathway, just like pathways to cancer, where there may be a few druggable targets along the way. Mm-hmm. So what we're hoping for pathways. But I think there'll be so many that to say by looking at the DNA. In some cases, we will. I'm, helped have a letter in Science a couple of weeks ago, which said basically we should sequence personal genomes from 100,000 people. Now that's saying, the cost will be down to $1,000. The cost will be nothing. The interpretation will be expensive and long running. And we'll need that to sort of search for the pathway. So, wanting to do 100,000 genomes is just, to me like arguing for motherhood. You shouldn't, just do it. And do it as fast as possible because, so, we've got the $1 billion for the cancer genomes. I think for 100 million we can come forth with the brain ones. And
do you think this is largely a question of money raising or consciousness raising or price lowering?
Oh, it's price lowering. I mean, that's the wonderful thing.
Why are you so sure of that? If I lowered the price of shaking hands with a tarantula to 0, you might not want to shake hands with a tarantula.
No, but just getting these genomes, I mean, if you don't want it, it's immoral. I mean, mental, if you really live with mental disease, as I do, it's not easy. And you just want to do something about it. And we swept it under the rug. Only Meryl Streep has come forward and says, that there's lots of bipolar in her family. I had a nephew who died of it. It's awful.
And you're pretty convinced that out of this kind of science will come therapies and cures, and therefore everybody should want it eventually?
Well, given the way science medicine goes, it'll probably lead to some therapies. And I think pre-implantation diagnosis will be very important for families with bipolar disease. A woman has bipolar, and she doesn't want to have a daughter who is. So that's going to be done. They're beginning to do it for BRCA1. You could argue, which is, well, I'd say, being bipolar is much worse than BRCA1. But, you can argue that because of, that's one of those things you can't decide. But they're both nasty. And, but they're beginning to do pre-implantation diagnosis for BRCA1. And, if I were Jewish and I had that thing, I wouldn't pass it on to, a son or a daughter. It's awful. So I just get rid of it.
So let me ask you one last question so we can get on with others. But are you amazed that from sort of 1953 going forward, or from 1968 going forward, that we are now at a point where we can do fairly, and that's really one of the questions for the room, fairly intimate investigations of people's inheritance?
I'm very happy.
Yeah, I know that. But are you?
But did you
happy that we can do it. I never thought we could.
You didn't think when you were working on the double helix, you didn't think someday we'll
I
read the
didn't
whole
even
thing?
think of it when we finished the Human Genome Project in 2003. I mean, until we got next generation sequencing, it wasn't worth thinking about. I mean, I'm not a technologist, but once you got it, I think it's, the sciences, Curtis, is so wonderful. Because we don't have to talk about money. It's just how, whether the human race is bright enough to get its act together. And, we did get our act together for the Human Genome Project. And, most people were against it. So, and, you hear this, ethical stuff, which is just crap.
Oh, come on.
Yes, it is. I mean, it's a sort of
sit down, then.
No. I'm really saying it's a minor concern. It's a blip on the whole spectrum of mental illness.
I just want to ask.
That's all I'm trying to say.
I just want to ask from here, is it just a matter, well, you were talking about figuring out pathways and so on to use this information from the genome. Are we just talking about just sort of industrializing biology, or are there going to have to be like really new concepts that people come up with that unify what we're finding out?
The limiting factor now is the intelligence of the scientists, not money. And it's just marginal. I mean, for what we're trying to do, because, you know, I've asked why, why we haven't learned more over the last 2 years. These genomes have been done, but they're hard to look at. And, Esther, and, I want to know why you haven't put your genomes up like I did. Because, not one of your people, you haven't done it, George, you're guilty. Put it on the web. What? Oh, okay. Not very long. How long? I'm just saying, until they're up there, you should encourage people, who, send them a pie or something like that, if they put their genome up on the web. And because until we get them up there, until we can compare them, we can't do phenotype and genotype. So, we'll need, again, a very large number of people to put it up. We can't do it with 100 people. That's, we'll settle a few things, but for the whole, we'll have to have it up. And there's going to be some, cases when I think we've got to be pragmatic about it. And when I first said it, I think we should just, take everyone in Esther's and reach, my age of 80. And then when you're 80, you can put it up because you don't really care, And even your kids have, got to the point where, that it's not as important. But
we will go on now, and we will go start talking to some of the younger folks. But
Yeah.
Thank you very, very, very much, and we may call on you again.
I want to, thank everyone for, making possible the sequencing of my genome, which controls now my blood pressure. Thank you.